Hello, I'm Barbara from Mapper Research. Is a society located in Area Science Park of Trieste. And Mapper Research develops and produces a lot of things, and principally instrumentation for nanotechnology. And our main products are scanning probe microscopes, SPM, and nanopositioning systems and sensors. SPM includes three main type of instrumentation of microscope: STM, AFM, and SNOM, or scanning tunneling microscope, atomic force microscope, and scanning near-field optical microscope. These SPM are the basic instrumentation for nanotechnologies, and probably the main SPM technique actually used in biology and medicine is AFM, but also SNOM is suitable for nanomedicine application. And we in BioNano Lab we applied this technique in particular for biomedical application. Here we can see one of the instruments that we use and produce, and is a SNOM with a biological setup. SNOM is a particular SPM technique because have a particularity in the probe. The probe is an optical fiber, so this technique can combine the SPM topography, like in AFM, and the optical information from the sample. And more, this optical information is presents a very high resolution. With the near field, we can overtake the diffraction limits, the limits that have all the traditional, conventional optical microscopes. This is a schema of the working principle of SNOM. And in SNOM, we create, in our SNOM, is a SNOM in aperture. In this case, the tip is an optical fiber that is sharpened and metallized at the end. And at the end of the tip, there is a hole, and the aperture is smaller, so smaller that is smaller than the wavelength of the light that you use. So the in this case, the light, generally laser light at different wavelength, the light that is coupled into the fiber, at the end of the fiber can pass through this aperture, because this aperture is smaller than the wavelength of the light. So the light can pass through as a classical far field, but create a so-called near field, o luce di campo prossimo. And so when the near field interacts with the surface of the sample, we can interact and scatter again as far field, and we can collect this far field as reflected or transmitted light, or eventually as a back reflection light into the fiber. Actually, Mapper Research is a partner of a multidisciplinary network created in a project for SPM application in nanomedicine. In particular, actually, we are working in collaboration with different research groups of Area Science Park, of laboratory of CISA, neurobiology group, and with the Department of Medicine and Physiopathology of University of Trieste and University of Udine. And today we are working principally on a project that study the cell membranes morphology and the interaction involved in different kind of disease. In particular, first we are working on oligodendrocyte culture involved in some disease, in particular in multiple sclerosis, because oligodendrocytes are the cells that create the myelin layer, and the myelinization is the process involved in multiple sclerosis. We are working on analysis of these oligodendrocytes with a different type of microscopy. We are working on comparison with SEM and other optical microscopy, traditional microscopy, like fluorescence microscopy, with SPM technique, in particular SNOM technique. And this is some preliminary results. We compare here SEM images of the branch and the elongation, different type of elongation, lamelliformi and lamellipodia and filipodia of oligodendrocytes. And we are studying this branch with the SEM and with the SNOM. In this case, with the SNOM topography, we can have achieve a higher resolution in topography, and we can measure exactly all the structure at the nanoscale level. And but we see with the SNOM you can have also an optical information. In this scheme we can compare different technique, optical technique. With the conventional optical microscopy, generally you use dyeing or fluorescent or not fluorescent dyeing. And but there is a limit, diffraction limits of the light. And with confocal, you can achieve a higher resolution, but also you have a problem to dyeing and labeling the sample. With SNOM, you can have more resolution that overtakes the diffraction limits, and also you can use specimen with marker or without any marker and without treatment. Here there is some other example of imaging that you can have with the SNOM. You can see there is an optical conventional microscopy. You can see the branch of this oligodendrocyte is not so visible because the sample is not marked. In SNOM topography you can see all the branch, and you can have also an optical reflection and an optical transmission images. And these different type of optical information give different information because the working principle is different. You can see in this scheme the difference between reflection and transmission is known. you can obtain a non-transmission optical image when the light passes through the entire sample, a cell or a section, and in this case the image is more similar than the conventional optical microscopy, but you have a different type of illumination. In conventional microscopy you illuminate all the sample simultaneously. Here you illuminate the sample point by point with a near-field point source, and in reflection, instead in reflection image, the information is due only from the reflection of the layer, a small layer under the surface of the sample. And the thickness of this layer depends on the type of the probe, and in particular depend on the aperture of the fiber, optical fiber that you use. And so in reflection image you can characterize only the structure that are below the surface. In our study, in particular, we are working on a procedure and on a method preparation of the sample. And we also applicate some procedure to mark this oligodendrocyte. And in particular, we start from a classical immunolabeling with antibody against alpha-tubulin to study cytoskeleton, and then we marked with DAB staining. And this method was developed and optimized for SNOM technique. And here we have some preliminary results of the study of this procedure. In this case, you can see the same type of cell, oligodendrocyte, and cytoskeleton with a conventional optical microscopy, with immunolabeling and with DAB. You can see the fluorescence imaging. And under you can see SNOM images. You have topography and reflection. Reflection in this case is obtained with this new method of preparation of the sample. And actually we are working on extending these methods preparation to other type of cell. In particular, we are working on other neurons, spinal ganglion neurons, involved in some disease, in particular in migraine. And also we are working on study filter of glomerular renal. And because we are studying this filter involved in some nephropathies and glomerulosclerosis. Now we are working also in some other application using fluorescence marker. So, thank you.
Thank you, Barbara. So now Roberta Carbone from Tetis will talk about nanomaterials from research to diagnostics. Thanks.
Okay, so good afternoon to everybody. Thanks. First of all, I want to thank the organizer of the meeting for the opportunity to present our activity in this occasion. The topic of today is about nanomaterials and especially related to the performance and the importance or the relevance of nanomaterial from research to diagnostics. Briefly, I will outline my talk with some introduction by defining a little bit nanomaterials and their potential application in different fields. Then I will try to give you some hints regarding the technology for nanomaterial production that we have in our company and describing a little bit of features and properties. The second part of my talk will be actually related to the description of biomedical application of nanomaterials, especially in research and diagnostic-based application. At the end, I will try to give you a brief summary to underline the main point of my talk. First of all, I want to just briefly to say that nanomaterial are really broad kind of material. They are nanotubes, nanoparticles, and of course nanostructural material, thin films, and so on. But what is important to say is that they can be distinguished from a point of view of their synthesis, their deposition. We can even define the construction of this material as a bottom-up construction. It's the method through which the nanobuilding blocks, that we say atoms, of the material can be assembled and produced by itself, going together, a nanostructural material. This is one technology to produce nanostructural material. The other one that is completely different can be called top-down technology of top-down construction. It means that using sophisticated technology like photolithography, you can fragment the material like a fine sculpturing from bulk material, like you see that here, and obtaining fine nanoscale structures. As example of this kind of deposition technologies or synthesis technology, I just put here to example, just for clarity of description. We have here a nanostructure thin films where atoms build nanoparticles, and then nanoparticles on top, one after them, they can build a layer, also thin films, in a, let's say, self-assembled model. On the other hand, we have a nanostructure silicon wafer, maybe you know very well, in which chemical, physical, or even mechanical approach are used to build nanoscale feature like this. So they are two different approach. You can call them nanostructure material, but the property of this material are different. And of course, what is very intriguing and interesting for people working in biomaterial and in biomedicine, the self-assembling approach that's at the bottom-up construction is attracting now a lot of interest. Okay, so the potential areas of nanomaterial application is really very broad. Of course, there is no time to go into detail of all this field. I want to just give you the information that TETIS is now focusing the activity in the biomedical application is, of course, is very broad. And in particular, we are dedicating our effort to medical diagnostic applications. So we are dealing with diagnostics devices. And today I will give you some hints and some results of our latest work. Now, some words related to the production methods that we use in TETIS. Of course, I will not give you the details of this deposition technology because are very complicated, but we produce nanopowders by flame spray pyrolysis, a very complicated system, and coatings or thin films by a proprietary technology that is called supersonic cluster beam deposition. Actually, both technology are considered to be bottom-up technology because from atoms, we can start from atoms, build nanoparticles, that with one technology we can produce powders or nanoparticles, and with the other technology we can produce thin films, like you see in this slide, coatings and thin films or different features. What is important to say is that the technology we have developed try to solve some of the main bottlenecks that are present in the industrial use of nanomaterials, such as the capability to scale up the production. And this is an important point if you want to go toward the production, of course, toward the market. Then also this deposition technology are completely compatible, easy compatible with the integration, the integration of these nanomaterials are very easily integrated into devices. So we can combine microfabricated devices into this deposition system and produce coated devices, micro devices. Then also there is another property that we can use different range of materials to produce either powders or layers or thin layers or coatings. So now some features related to the nanostructural coating. And these are features mainly related to the biological and biomedical application. Here you have some example. This is an atomic force microscopy image of a titanium dioxide thin film, and this is an electron microscopy. And what you can see very well from this picture, that this kind of nanostructural material possesses high specific surface area due to the nanoscale roughness and porosity that can be finely tuned according to the thickness of the layer you want to produce. And this is an important parameter for the interaction with the bio world. Then this material presents a very good adhesion to on glass. So you have a layer of a thin film on a nanostructural material perfectly adhere on a coat, on a support like a glass. And so you can use, of course, in biological application. Then most of them are optical transparent, and they possess also low autofluorescence background. So this is the very important property if you want to go toward an analytical assay that involves immunofluorescence readout. Moreover, this deposition technology is compatible, is performed at room temperature. So this provides compatibility with any kind of substrate, like plastic. So you can produce plates or other devices in plastic and coat them with this kind of nanostructural materials. Then also you can do another nice things, avoiding photolithographic processes to produce patterns that are very different geometry and very tiny. Like in this example, I don't know if you can appreciate, here there are small dots of nanostructural layer, thin layer of nanostructural titanium dioxide, like an array of dots on a glass slide. So we can do this by using a hard mask in front before the substrate, and nanoparticles goes inside of the dots and then go to impinge on the substrate and remain there. Then at the end, as I told you already, we have a wide group of synthesizable materials like noble metals, simple oxides, complex oxides, and composites are available for this kind of synthesis. Then now we go to the biomedical application that we have exploited so far. We have started to study the interaction with the different biomolecules like DNA. In this case, this is an internal research activity that's ongoing. Then we have also demonstrated the biocompatibility of different kinds of cells with nanostructured titanium dioxide. And also unravel a very interesting property of triggering adhesion and also immobilization of cells, different cells. Then recently we have just accepted a paper for the usage for the utilization of this material as efficient binder for proteins and peptide in protein macroarrays assay. And at the end, we have also shown that also complex systems like retroviral cell arrays that are really very complex, and I will not go into detail for the description, they can benefit from the property of nanostructural thin coating, thin layer material. So far, this is something that we have done in a research setting. Of course, we think that these are important property to go toward diagnostics and toward the market for diagnostics. And then we are now moving forward to trying to integrate this material into devices that can benefit from miniaturization and possibly automation to create improved diagnostic device with novel added value for disease monitoring. Now I give you an example of what I was saying before. If we have non-adherent human peripheral hematopoietic cells, these cells do not adhere, are not immobilized very easily on surfaces. They are on blood, so they are usually going on the blood, so they are not used to be adherent. So we have uncovered an unexpected property in which a nanostructured titanium dioxide, as you can see in this picture, trigger a sort of immobilization of the cells once you see the cells on this material. If you compare the morphology of the cells with the other one, this is an uncoated cover slip, this is a coated cover slip, you see the morphology is completely different. So the cells are immobilized on this material. Immobilization is a prerequisite step for many different analytical assays that can be important in diagnostics. And also an important point to remind that we can integrate the coating in microlaboratory devices. So the step is very short because we can go toward a production, the creation, and this is something that we are doing right now, of a nano-coated microfluidic slides that can be used for cancer diagnosis, especially for hemato-oncological disease. And I will briefly explain how it works. So we can coat a microfluidic channel with a nanostructured titanium dioxide, then load the hematopoietic cell even from bone marrow or peripheral blood. The cells undergo immediate immobilization on the surface, and they resist shear stress that is produced by all the reagents you can add in an analytical assay. And this is the result. I don't know if you can appreciate DAPI staining, maybe it's too dark, but on the nanostructured titanium dioxide coated microfluidic channel, which size is 1 centimeter long and 300 micron wide, you can retain more than 80% of the cells on the microchannel. And these cells have undergone a sort of series of steps of reagents like fixation, permeabilization, labeling, staining, and so on, and they stay there. While in the uncoated glass, only 10% of cells are left. So this kind of device can be really very useful, and we are now in test with 2 hospitals for biomarker detection with fluorescence readout, in particular for the analysis of DNA and chromosomal situation. This kind of assay can be, of course, is a miniaturized assay. It can be automated and offer, of course, the following consequences. You can have a dramatic decrease in reagent cost and sample amount. So you can even think to provide this kind of device for molecular screening of specific kind of biomarkers or can be of interest. So at the end, just a brief summary, what we think that nanomaterials are really relevant, that can provide improved functionality for research and also diagnostic application. The integration of nanomaterial in this device, that we have just give you an example of with this kind of chip, and that through miniaturization and automation, this kind of chip can really become a sort of important tool for molecular screening of biomarkers. Now TETIS is capable of entering the diagnostic field on specific cell-related disease with this kind of chip using nanomaterials integrated into the device. And I have to say that this plan is now almost at the end of the pilot study, so we are starting the procedure for the certification of the device, and we hope that the next year we can enter with the field, let's say, of the research market with this kind of device. I want to thank at the end all the team that have participated to this project, and especially Andrea Marzi and Samuele of the Biolab group, and Emanuele Barborini and Simone Vinati that are responsible, let's say, of the group leader for the applied research and system and apparata. And thanks for your attention.